A very good afternoon to all the interested audience who have joined us and who are about to join us for the second talk of this Dasara Lecture series. As you all know, today we will be having a talk by Dr. Vijaykumar Krishnamurthy on the size and scaling laws in living systems. So without taking much of your time, I request our director to welcome the speaker as well as the gathering.
Excuse me. Good afternoon to you all. I welcome you all to this second lecture in the Dasara Lecture series of this year 2023. Yesterday we had a lecture on distance ladder in the universe, which was interesting. And I'm very sure that today's topic also, the size and scaling laws in living systems, will be as interesting as yesterday's, and perhaps more even. So we are all part of living systems, isn't it? So that's the way. So to give this talk, Professor Vijaykumar Krishnamurthy, I understand that he was associated with this institution and he is very proud of it, and I talked to him and I was also very much impressed with his simplicity, and he has been he has kindly agreed to speak to the audience here on the occasion of the second Dasara lecture, and I don't want to interfere in this regard, and without wasting much time I would invite Ms. Lakshmi to announce the next topic. Thank you.
A small introduction of the speaker. So, Dr. Vijaykumar studied BSc in MES College, Bengaluru, and he did a PhD in physics at IISc, Bengaluru. He spent a few years as a joint postdoc at Max Planck Institute for the Physics of Complex Systems and the Max Planck Institute for Molecular Cell Biology and Genetics, both in Dresden, Germany. He returned to India in the year 2015 and joined the faculty of ICTS. He says that he never liked biology in school and left it as soon as he could possibly do that. He also adds to that during his BSc days, the non-formal science education programs at JNP strengthened his foundations in physics and mathematics. Thank you so much for that, sir. Later, while studying soft condensed matter physics and non-equilibrium statistical mechanics, it naturally led him to ask questions on the physics of life, which he is currently pursuing at ICTS and plans to do so for the foreseeable future. So now I request our director to hand over a small memento to Dr. Vijaykumar, and I'll also request Vijaykumar to give his talk later on. Thank you, sir.
Thank you. Well, please. Thank you, sir. Thank you very much.
Thank you very much.
Better. Okay. It's a great pleasure to be back home. So this basically is the place where I trained in science during my BSc days. So I'm very grateful to the JNP team who have given me this opportunity to come and give a talk in the Dasara Lecture series. So like was just announced, I did not like biology in school. I left it very, very soon, after 10th. But then I went to physics and mathematics. Once you get a bit of training in physics and mathematics, the challenge of biology hits you in the face and sort of starts asking you to think about the physics and mathematics behind living systems. And that's the area of research that I pursue. I pursue one particular area of research in biophysics, aspects of developing organisms and cell division, cell and developmental biology. The size and scaling is not something that I do on a daily basis, but nevertheless, things do get connected up at a larger scale. So I'll try to give a talk which is aimed at a very general audience and does not assume anything beyond school level science and mathematics, but free to interrupt me at any point of time and discuss this. Okay, So here's the outline. So I'll start by asking questions about what is size, what is scaling, and how to think about size and scaling in very pictorial and geometrical terms. And I'll try to motivate that there are a large number of living organisms which have various sizes that we see in our natural world. And so I would like to understand, and I would like you to think about why is size important for living systems and how size is many aspects of the process of life. One of the things that will come up very nicely is how is this size or in general shape attained, and organisms develop starting from a single cell as zygote, and I'll show you several movies which will give you a feel for how organisms develop, how shape comes about in 3D space, and how this is very robustly attained in many creatures. One thing is to attain the size and shape, but the other thing is to be resilient to damages in size and shape. Examples being regeneration, something that we see in our everyday life. And I'll show you many examples of creatures which are very adept at regeneration. And, but there's poses very nice questions in mathematics and physics, because you need to understand how organisms know their shape and size. How is the mathematics of that encoded in the gene regulation patterns. Finally, I'll look at, if there is time, patterns that come up during growth and how they should attain certain scaling features to robustly develop shapes and sizes. My work mostly revolves around, and I'll give you some hints about the kind of things that I do, and I'll try to summarize. Okay, so I want to have a very broad background and try questions about what is meant by size, what is shape, and what is meant by scaling. And so since this is lectures on scaling, I'm sure you heard some aspects of scaling yesterday. But scaling has different applications in different fields, and I would like to set that out at the in very broad terms. So all of us know what is meant by size, typically, right? So it's basically the magnitude or the physical dimension of something. All of us have gone through many of the phases of, we understand that we grow. But when you say something is of a certain size, something is big or small, it's always relative to some reference object. There's some standard length that you're thinking about, and relative to the length, it could be big or small. That's how you compare sizes. There's no absolute size, so to speak. I'm sure you saw something like this in yesterday's talk, wherein you probably saw that the various scales that are there in astrophysical objects, starting from something like tiny planets all the way up to very gigantic stars. What is shape then? I like to think about this in very general terms. Surely the smallest kind of geometrical structures are points, and points don't have any shape. But nevertheless, objects like one-dimensional wires so don't seem to have much of a shape, unless you can bend the wire and look at it in 3D. But an object sitting on the wire itself will always feel this is basically a one-dimensional geometry, and so therefore there's no notion of very nicely defined notion of shape. 2D and 3D, there are very clear definitions of shape, and so you see them by trying to think about a representation of the external surface of an object, basically a boundary or an outline, and this kind of the overall geometry of the shapes and sizes that you would be familiar with everyday objects are shown here in 2D and 3D. How do I compare shapes? I mean, does it even make sense to ask, is this shape the same as that shape? And so one would like to ask, in what sense are you trying to compare? And this is something that we've all learned in school, and I want to recapitulate that and put it in a firmer footing. So there are different kinds of ways in which you can compare the shapes of objects. So there are objects which are congruent to each other. Basically, you say that if I have a red square and a blue square, and I ask, are the two squares the same shape? One way to think about it is, you can, if you take the red square, and if you can identify it with the blue square by doing translations and rotations. No size changes, only translations and rotations. Such objects are congruent to each other. Something that you learn in school as congruent triangles. There's another set of transformations that you can do, similarity transformations. So in addition to translations, rotations, and reflections, suppose you're also allowed to do scaling, change the overall size of an object. Something that you also learn in school, similar triangles. So there's a similarity transformation, and the similarity transformation also gives you a way to identify different shapes, and so therefore to compare shapes. A more general class of transformations are what are called homeomorphic transformations. These allow not just changes in size, rotation, translation, but also deformation. smooth deformation. So you basically have to think of like a of clay, which you can mold into any shape. For all such shapes are different to each other. So if you can identify two shapes by doing not just translation rotation, but also deformation, pairing allowed, homeomorphic to each other, they're also equivalent. So asking questions about whether two shapes are equivalent or the same really depends on what is the metric that you are using, what is the measure you are using to think about comparison. So for instance, in this picture, all objects with the same color are similar in shape. They're not congruent in shape, they're similar in shape. But if you extend the equivalence of shape by homeomorphism, all of them are equivalent. So when you say how different are shapes or how different are sizes, so you need to think about how is it that you're thinking about shape and size. So one thing that I introduced very sneakily here is the idea that there is something called scaling. That is the idea that you can take an object and you can either expand it or shrink it, and yet you consider somehow that object to be the same as the original object. So I want to make it clear. So think about a rectangle, some width and some height, and let's say I look at another rectangle with a different width and the height, and if the ratios of the widths is the same as the ratios of the heights, in other words, the ratio of width to height is the same as the ratio of the width to height for the other triangle, these are similar rectangles. And so therefore there is the notion of proportionate scaling. So you have scaled out the rectangle proportionately in all directions at all points on the rectangle. But that's not the most general transformation that you can do for scaling. So in general, you could do a transformation where the width and the height scale differently. So this will be the most mathematics that will be there in the slide. So if you're scared of that, don't worry too much about it. So basically the idea being that you can have a width which goes as a power law of the height. Familiar examples could be, for instance, area which goes as the square of the length of the side. In such a scenario, you would find that the ratio of the widths is the ratio of the heights to the power k, some scale. So the usual scaling that we learn in school when you think about similar triangles and so on is the case where k is equal to 1. That's proportionate scaling. Typically when k is not 1, the exponent is not 1, scaling is also associated with shape changes. For instance, if I have a circle, and let's say I scale the widths by some ratio, but the heights by a different ratio, then the circle gets transformed to an ellipse. A circle and an ellipse are not congruent, and they are also similar, though they are homeomorphic. General pictures that will come up when you think about scaling laws are relations of the following kind. So let's say you have some variable x, and there is another variable y, and the dependence of y on x is something like a bx power k. Some exponent need not be an integer, it could be some rational number too. So the study of such laws in living systems is what's called allometry. So basically you want to understand how various properties of living organisms scale, for example, with their length, with their body mass, with their area, and so on. So for x, for instance, could be length, mass, or even time when an organism is growing in time. Y could be things like area, volume, maybe how long the animal is alive, maybe how long it takes to regenerate it, maybe some measure of the internal complexity of the organism, or maybe even something like the metabolic rate, how much energy it consumes per unit time. Many such quantities will be related to typical size measurements via a power law. Something to learn when you think about such power laws, because we discover such power laws in experimental data by looking at plots of y versus x, but on a log-log scale typically. So if you have y is bx power k, think of k as 2, so y is bx square, that's a parabola. A plot of y versus x on a linear scale will give you a curve of this kind, something that everybody should have seen. But then to discover that it's indeed a power law, you'll basically look at the plot of logarithm of y in any basis and versus logarithm of x. And in such a scenario, if y and x have a scaling relationship, the plot turns out to be a straight line. Much easier to detect in experimental data. So you would plot log y versus log x, and if the straight line the data appears to follow some sort of a straight line, that's a very good indication that there is indeed a scaling relationship between the variables y and x. The slope of the straight line in the log-log plot is related and is exactly equal to the exponent b, and the intercept of the straight line is the prefactor B sitting in the scaling law. So something to keep in mind, because such plots will appear again and again, and one have to keep a sharp eye on whether you're plotting something on a linear scale or a log scale. So typically you assume that you have good scaling only when something scales linearly in a log scale. So if this exponent k is greater than 1, it's called positive allometry, so which means the variable y scales faster than the variable x. Conversely, if k is less than 1, the variable y scales slower than the variable x. So examples of positive allometry, for instance, if x is the length and y is an area, the area is length power 2. So that as you scale the length by a factor of 2, the area goes up by a factor of 4. So these are important in biology. So here's a bunch of hypothetical butterflies, and I want to use this to illustrate how to think about such scaling plots. So here are plots. In the plot on the left, you have butterfly sizes, let's say the wing size as a function of the body size of the creature. You find that there are two kinds of straight lines. These two straight lines, if you observe them, remember on a log-log scale, have the same slope. They are tilted the same way, but they have a different intercept. So the intercept is basically measuring this proportion factor B. And the slope being the same tells you that the exponent k is the same for the two features. So if you have differences but not the slope, this sort of tells you that the proportionate size, in this case the wing, scales irrespective of the body size. Another plot where, let's say, you keep the same proportionality factor B, but have different k, the slopes. So in this case, you see that as you change body size, the wing size goes much faster for the pink ones compared to the light blue ones. So if you have differences in slope, k, then this sort of tells you how the relative size changes as you change the body size. These measures can be used to compare sizes and scalings both within species and across species. Okay, so there are size laws and there seem to be scaling laws that we're thinking about, but I would like to understand there are in living organisms, something that we all know about, and how do I think about such size variations and how do I actually quantify them? So here's a classic plot from H.G. Wells and Huxley 100 years ago. It's a beautiful plot which tries to illustrate the relative sizes of the organisms that are familiar to us, who appears somewhere here and somewhere over there, just to give you a size. So you see the largest creatures are really large, and that's the blue whale, and the human is sitting inside the blue whale to see a size comparison. This is a sequoia tree. It's probably the largest living creatures that we know on Earth. At the same time, there are a bunch of creatures which are extinct. There are a bunch of them which are around now, and a huge range of size variation. That's what the plot is. So there are snakes and dinosaurs and so on and so forth, but hidden in this is something very, very weird. Do you see the straight line just below the snake? That's actually a tapeworm. Actually, it's supposed to live inside humans. Oops. So you have something far bigger than a human living inside a human. That was size variation across species. But you seem to have size variation even within species. So here are two dogs. They're both dogs. They're the same species. Look at the size comparison there. One is massive compared to the other. A very interesting thing to, how is it that species can also show such large variations in apparent size? It's not just in non-human species. People have speculated at size variations even in humans. This is classic tale, I'm sure many of you would have heard, Gulliver's Travels. Gulliver was a very nice explorer. Takes a ship and goes around some very special island, finds a set of people who are very, very tiny, the Lilliputs. So this is Gulliver, and this is the Emperor of Lilliput addressing him. But then he spends some time there. There's a very nice story that develops. After some time, he gets away, he gets picked up by a ship, and then he gets marooned again, or maybe he goes around another trip, and the next time he lands up with a different island, Bob Dinglang, and there it turns out that Gulliver is the Lilliput. So there are creatures, there are humans who are huge, massive compared to Gulliver. So such speculations have been around for a very, very long time in the human imagination. They're not out of context. So recorded tallest person that we know is shown here, Robert Wadlow, and he scaled close to 9 feet. And this is his father, who's an apparent average-sized human being. So it's not even true that the same species or even related species can have the same height. I mean, you can have really various heights there. Many of you might have seen this. This was a video trying to show between the tallest person alive in 2014 and the shortest person alive around the same time. So that's a video showing the tallest person with an average-sized person, and there's the shortest person around there too. So if you look at the scales, there's a factor of 5 difference between the shortest person and the tallest person. 50 centimeters, 150 centimeters there. Some things to understand and observe right now. Look at, for instance, the sizes of the heads or the internal organs, hands. There is proportionate scaling. So that's something that will turn out very important and a problem that we don't understand very well yet. Is it that as the size scale very, very proportionately? One thing is to look at adult organisms and then ask questions about the sizes of these organisms. Variations and size scalings even in precursors to adult organisms. For instance, birds and insects lay eggs. There's a huge range of egg sizes. Recent studies have actually characterized shapes and size variations in birds. So, like I mentioned to you, this is a log scale, and you see some large scale variation in the ellipticity and the asymmetry parameter there, indicating certain scaling laws between ellipticity and symmetry. These are insect eggs. Insect eggs are far, far varied in shape and size, and you see a scatter plot of the insect volume to the aspect ratio, the egg volume to the egg aspect ratio there. And even the shapes of the insect eggs are very, very varied. To play this movie, you will see a movie starting from the smallest. We scale out the size variation there. So that's a, and as you go up in size, the insect eggs vary in size to a very large extent. So it's remarkable that there are insects who lay eggs, and the egg sizes vary by a very large. Why is it important? How do we understand? I mean, why can't there be any arbitrary size? I mean, most of us here are roughly the same size, maybe 5 to 6 feet. Why can't people 100 feet tall? Or why can't we have people 1 centimeter? I mean, is there a size that is set by typically the kind of structure that is there in the creature? And so if so, does size also determine the structure that comes along? Size is controlled and also affects certain features. So for instance, size very strictly controls the strength of an organism. So it really depends. For example, if you're very tall, your bones will break at some point. You'll not be able to bear your own weight. You basically have to reduce your weight, and at some point of time, you'll be able to balance the pressure on your bones. It also affects the food intake rate. For creatures which have to absorb food from the surroundings, the surface area is the primary determinant of how efficient they are at getting food. And so the surface area also goes up with size. And so therefore, depending on the environment that they're in, the surface area will also restrict kind of sizes that can come up. Perhaps something not is division of labor, basically the complexity, the internal constitution of living creatures, and I'll come to that in a minute. But not just that, various rates, for example, rates of metabolism, how long it takes for the creature to generate, starting from an egg, how long does it live, and how fast can it move. All these seem to be dependent on typically the size, and size seems to set a kind of scaling relation. But even at a larger scale, even at the ecological scale, the abundance of an organism seems to depend on its size. Well, if the organism is too big and it needs a lot of food, there isn't enough food in the environment, it will die. If it cannot feed on the other creatures, but it will die because the other creatures will eat you up. So there's a dynamic competition between the size and the environment that we're living in. This sort of sets the size, but it also sets the ecological abundance of the organism. So I would strongly recommend you read this book. It's a reasonably non-technical book by John Bonner, which really puts out many of these aspects in a very beautiful fashion. So like I was mentioning, there are various rates about taking in food, metabolism. Here's, for example, a classic study that many people have been doing for the past 100 years. This is one recent paper that I picked up. You try to look at, remember, log-log scale. So you try to look at the metabolic rate, the rate at which you consume food and dissipate it, as a function of the mass of the creature. So like I mentioned to you, you have a log-log scale, and if you have a straight line, that means the metabolic rate is the mass power something. And that exponent has been, which has been for a very, very long time. I don't know, Professor Ganesha might touch upon these things on Sunday. But if you look at the size of organisms, let's say at the present day, there's some size variation. We all know it began about 4 billion years ago, 3.5 to 4 billion years ago. Has the size distribution been the same throughout evolution? Or have there been major events where the size distribution drastically changed? So a recent study pointed out the following. So they made a compilation, a largest compilation of all the largest known fossils, fossil records there. And they plotted the size, volume here, function of how old the creature was in terms of millions of years. So you find that starting from the earliest known living creatures to the largest known ones in the present day, there's a 16 orders of magnitude change in the volume. So which means if you take the volume of the largest creature now, divide by the volume of the tiniest known creature in the earlier days, the factor is 10 power 16. This is 1 followed by 16 zeros. That's the huge volume that we have encountered in life. But then if you look at the plot, something that they pointed out and which is very, very remarkable, there seem to be discrete steps. So 1,000 to 1,500 million years is not varying much. Somewhere around here there's a sudden jump in the scale, and then it remains more or less around that value. And then suddenly one more step, and then it reaches something which is at the present day level. So I'd like to understand why, what are those major events? What happened around those times which actually caused a massive increase in organismal size? Turns out there are very important events in the history of life on Earth. The first event actually corresponds with the appearance of eukaryotic cells. Remember when we learn in school, we learn two kinds of cells. There are prokaryotes and eukaryotes. Prokaryotes don't have a specialized nucleus. The DNA is spread all over the volume of the cell. Eukaryotes have a specialized nucleus which holds the chromosomes and the DNA inside. And the eukaryotes are far more complex. They have much more internal organelles, and they use those organelles to do much more complex stuff. So the first jump in the size appeared when the first eukaryotic cells started appearing on Earth. Then there is another lull, and somewhere here is when multicellular eukaryotes started coming up. We ourselves are multicellular creatures. We're made up of billions of cells. And so the appearance of multicellular, or the emergence of multicellularity, dramatically changed the size and the shape of organisms. So that's the two major events in evolution which correspond to a massive change in size. I want you to keep note of this. This is again a log scale. But like I mentioned to you, the organismal size also depends on what happens in the environment. I mean, the environment can actually make the size go down or go up. So it turns out that these two steps also roughly correspond to the following two events in the composition of the atmosphere of the Earth. Here, the amount of oxygen that's there in the Earth's atmosphere was below 0.1% of the present level. And somewhere around the appearance of the eukaryotes, it jumped from 1 to 10%. There's a factor of a 10 difference in the amount of oxygen that's available. And at the time, multicellularity, it really jumped to the present levels of oxygen that's there. So there's an environmental factor, which is the amount of oxygen that's available in the atmosphere. And that also kind of controls the maximum sizes that are possible in living organisms. Remember, there's a feedback between the two, because the kind of creatures that come up can also consume oxygen or give out oxygen. Plants will dramatically increase the availability of oxygen. So there's strong coupling between the kind of complex structures that can exist and the environment that can support such structures. Okay, so this is about trying to understand that there are organisms and their sizes seem to vary across within species. Think about the variation of those organismal sizes as a function of their linear lengths and scaling laws. But then many organisms, particular ones in particular, including us, don't at this size, at the adult size. We start out very, very tiny, not even at the baby level. All of us were once upon a time a single cell inside our mother's womb, a single cell. Now we have 10 to the 12 cells. How did they come up? How did the size come up? How did the shape come up? That's something which is very fascinating to understand. So what I wanted to do is to show you a bunch of movies, these organisms that develop, and to give you an appreciation for how size and shape comes up very naturally during the course of development. So this is bacteria, this is prokaryotes. So there isn't complexity here, it's basically a cylinder. And there's a wire of DNA floating around, a bunch of gooey proteins. And at some point of time, the cell decides to divide. And so if you watch the movie, one bacteria becomes 2, 2 becomes 4, and so there's an exponential increase in the number of bacteria over time. And what you get is more and more bacteria. These are not multicellular structures. Now, this way of making copies of yourself, which is the fundamental tenet of life, cell division, is very simple in the case of prokaryotes. It's far more complex in the case of eukaryotes, much like us. So this is a eukaryotic cell in some kidneys of pigs, I think. And what you see is the event where a single cell divides, and the division of the cell is accomplished by a bunch of tasks. The red is mostly DNA, some fluorescent labels. The green is a certain set of polymeric filaments called microtubules. So the microtubules actually this spindle. So you see this very nice spindle-like, green-like structure that comes up. When the cell decides to divide, the chromosomes duplicate, learn in school, and there are 2 sets of chromosomes. The 2 sets of chromosomes are pulled apart to the 2 sides of the cell. It's mitotic spindle. There are questions. How does the size of the spindle itself scale as you get bigger and bigger cells? I'll come back to that sometime later. But this is single cell division. Surely we are multicellular creatures. And multicellular creatures have, by definition, many cells. And many cells somehow conspire together to give you shape. So here's a dramatic movie. So this is the development of a zebrafish, starting from a single cell. So this is the zygote, this is the fertilized egg. You can take it out of the shell, put it under a microscope, make a movie of it. And if you maintain the temperature, pH, and the environmental factors very similar to what sits there in the eggshell, the creature is very happy to divide and develop into a full-grown fish. So this is yolk. This is a bunch of cells that the zygote is available until it can become big enough to fend food for itself. So look at the scale here. This is about 0.5 millimeter in size. And this is hours, minutes, and seconds. So if I run the movie, start seeing a single cell dividing into 2 cells, 4, 8, 16, and so on and so forth. And at some point of time, the cells not just stop dividing, but they start reorganizing. At some point of time, you start seeing the appearance of familiar structures. Fish are vertebrates, so you should see the appearance of a backbone that we all have as well. At some point of time, you see the structure of an eye, pigmentation. This is something that can be seen in the lab. So you have not only an increase in size, number of cells are going up, the mass is going up, but you also have a change in shape. How does this change in shape come about? I mean, how is it that a single cell knows to become a fish? Big open question, we don't understand the physics and mathematics behind this. Here's another example. Before I go to the example, the general trend that seems to be in multicellular creatures is the following. So you typically start by a single cell. The cell is fertilized by the entry of the sperm. It forms what's called a zygote. And then you see a huge number of divisions. That's called the cleavage part, where you have an increase in the number of cells, but the mass remains the same. And after that comes the process of gastrulation. This is basically the process where the cells start rearranging and start getting in the shape of, let's say, a tube, tubular structures. And then comes differentiation, which is basically specialization. So you maybe become heart cells, eye cells, and neurons, and muscle cells, and so on. And eventually, at the shape of a creature. So keep track of these changes and let's see them play out, for example, in frog zygotes. So these are 3 frog embryos. They are isolated. You can see a thin shell around them, the white background. And so they're suspended in it and they're not talking to each other. They're more or less isolated. And when you run the movie, I would like you to observe this dramatic sequence of changes that happens from a single cell, multicellular structures, rearrangement of the multicellular structure, and eventually the emergence of a familiar shape. So there are cells, and the things that you should also ponder about is the synchronization in the divisions that's happening there. So 1, 2, 4, 16, many cells. The cells start rearranging. They folding upon themselves, the backbone-like structure that comes up. At some point of time, a tubular structure has appeared. You should see the formation of eyes, the internal organ systems, and the familiar dramatic thing that happens every day. And we have no clue how it works. There's a fantastic movie that went viral on YouTube a couple of years ago. This is trying to show you the development of a newt and of a salamander, and a really very gorgeous movie with very high resolution. So again, you see it's in an eggshell, familiar process whereby many, and the high resolution movie will start showing you, for instance, the way the cells start invaginating. They form a tube-like structure. At some point of time, that's the tube that forms there. There's a massive sequence of shape changes. You should really be thinking about all the homeomorphic transformations that we are thinking about. And at some point of time, you see the formation of a head, a backbone-like structure, and a tail-like structure coming up there. You see a huge amount of movement of material, cells, some rudimentary form of blood. Some point of time, start twitching, acting. This eventually leads to the formation of the heart, blood pumping there at some point of time. You see a circulatory system is developing immediately. Familiar structures popping up. There's the heart beginning to beat. And at some point of time, a pigmentation pattern starts appearing, some coloration patterns on the surface of the creature. And there you go, a very well-defined salamander is formed, starting from a single cell. Happily swims away and tries to find food for itself. Okay? Okay, so there's a circulatory system, a higher resolution view of that. There's a heart pumping there. All started from a bag which was microns in size or 10 microns in size, a bag of chemicals. How that bag of chemicals changes size and shape and how it scales is something that really troubles people. Now, one thing is to generate size. Okay, there is size and the other thing is to generate size, but then you also have to be resilient to changes in size and shape. Classic example that all of us encounter in our homes. There's a lizard and you suddenly scare it and it drops its tail and runs away. And you're focusing on the tail and the lizard runs away. It's a very nice ploy, right? So your attention is diverted to the wiggling tail there. So this is something that the lizard does it very cleverly to evade predators. So it's called origami. The tail is not lost. Maybe if you find a few months later, you'll find it has grown a full tail. Does it know how to grow back and get the same shape and size of the tail? So this process by which they sort of are resilient to environmental conditions is what regeneration. So you can ask questions about, is the regeneration complete? Do they fully recover? Maybe there's partial recoveration. Do you regenerate the size and the shape that you had at the original time point? Is it the original? Can you distinguish that? So let's study how these processes happens is regeneration. So it's basically defined as the ability of organisms to renew and restore lost tissues. It provides good resilience to fluctuations in size and shape. This is something which is very well seen in many creatures and something that people study very in the lab. So this is a newt, a salamander, and you can take a young newt, and what you can do is to chop off one of its limbs and then put it under a microscope and observe it. And over a period of like 50 days, the entire limb gets regenerated, both in size and in shape. That's a remarkable process. How does the creature know what size of a limb to generate? How size of a shape and the size of a limb to generate? I mean, you could have just forgotten the limb, but then it has an internal mechanism to generate that. So here's a movie that shows you that. So you have a amputation, 0, and that's the cut. And what you see is the subsequent regeneration of the limb. So this is basically a microscope movie without any labeling. And you see the mass of cells growing at the leading edge of the limb, and eventually a shape also getting generated. And towards the end, you should see the formation of even finger-like structures are getting. See that? So this is some chemical which is basically labeling all possible cells inside, and you see the chemicals are there throughout the limb, only the muscle cells. You see at the initial stages. When you go towards the end, the muscles also pick up. So there are various structures, and the various structures seem to go to the size of the organism. Another classic reason is to understand regeneration, and so therefore also aspects of size and scaling in living systems. Planaria. These are platforms that live in water, and you can actually take such creatures, observe their anatomy. It's a very simple anatomy, basically a tube for feeding and excreting stuff. Nervous system there. You can take such a creature, and in the lab you can take the creature and just chop it. So people would like to see what happens. Can it do anything? So here's a glass plate and you just cut it into pieces. This is super remarkable. It's like the thing of regeneration. You can cut it into 3 pieces in this fashion. In 14 days, each piece becomes a whole creature. You get the trunk, and you chopped up 3 parts, the head part, the trunk part, and the tail part, and you just give it enough food, and each part gets regenerated into a complete creature. Each part, internal system, and everything that's required to survive. Well, surely once you can do that, there are curious scientists and they would like to understand, suppose I cut it not like that, but maybe like that. Will it still regenerate? It does. Okay, maybe this fellow knows its length and breadth. Suppose I cut it obliquely. Well, it still regenerates. No matter where you cut it, the creature seems to be able to regenerate itself completely into a full body shape. Super remarkable. I mean, how is it possible that you take a piece of you and the whole of you comes out? That's just doing it right there. How does it even know what size to go to, what shape to attain, what functional organs to build inside? This is something which is really a subject of intense research in biology. Another classic example, Hydra. Hydra is a freshwater polyp. You will find it in many ponds here. And it's a hydrozoan. And so it has a bunch of tentacles and a main body. It's something where you can also get lots of regeneration. So I'm not showing the video. So people have done a chopping off of such a hydra and watched it under a microscope. And in a matter of several hours, a piece of the hydra becomes the entire hydra. So you start seeing a ball of cells, and some huge internal processes which are going on. And at some point of time, it starts elongating, and you start seeing these 2 pair of antennas coming up there. The whole creature is regenerated just from its part. Now, how does it do that? So there's intense research which shows there's a lot of physics and a lot of mathematics of the internal fiber organization in the hydra, and that seems to control how the shape and size emerges. Can everyone regenerate? Can you and I regenerate? And can everything be regenerated? So there are questions about this. So there are classic masters of regeneration in the living kingdom. So there's planaria, there's hydra, there are also certain lizards, salamanders, but there are also creatures which are like frogs. Frogs are very good at regenerating tails. There are fishes, zebrafish can regenerate its fin if it's lost, but something which is absolutely fantastic, it can regenerate its own heart. So even if it loses a heart, it can regenerate its entire functioning heart within a few days. Mice are very good at regenerating livers. We don't understand all these things, but studying them will give us lots of information about shape and size and scaling works in the living world. This is a evolutionary tree. So you can look at all the classes of creatures which are good at regeneration. So the ones in bright orange are creatures which have very good regenerative capabilities. Some of them we saw, like Hydra and Planaria and starfish and so on and so forth. There are a bunch of them which are marked in red, and these are creatures that can generate at least one body part. There are, for example, some insects can do that, some salamanders and so on. And there are a bunch of phyla, including us, which can't regenerate many body parts, or it's not very. So all this sort of assumes that there is a fixed body size, in the sense that if I chop off the limb of the salamander, it grows back a limb and it grows back to its original size and shape. There's no fixed size. So in fact, this is a classic example of planaria, where you can take the planaria, give it food, it grows without bound. It can become bigger and bigger, even of the order of a few centimeters in size. If you it just shrinks. How nice. So if you don't have food, you become small, you have food, you become big. You never die. How nice it would be. Planaria does that very regularly. But remember, these are creatures which are probably starting up from some internal single cell stage and then doing a multicellular structure, reorganizing it and so on. But there's also growth, in the sense that we on more or less with a good, and there is an increase in the size. We start going from infants to all the way up to adults. So when we grow, your internal organs also rise proportionately. As a tiny baby, you have tiny fingers. As you grow older, do you put on more fingers, or do your fingers become bigger? So there's proportionate scaling of the fingers. If the fingers remain the same size as when you were a baby, you probably would have by the time you are an adult. That's scaling of every internal body part as the overall size of the creature also grows up. How is this achieved? Remember, I told you early on that if you don't do scaling, shape will change. So that part don't change proportionately, the overall shape of the creature completely changes. That's not what happens to creatures. Basic example, we go back to Gulliver. So something to notice in these figures. Gulliver is big, but look at the Lilliputian land. The Lilliput king is small, but the mountains are also small, the trees are also small. So there is proportionate everything in. Similarly, in the giant world, there are bigger figures which are concomitant with the size of the humans that are there. Okay, does it work in the real life? It does. People can measure this in humans, in many different kinds of mammals. So for instance, in humans, again remember log scale, you see the size of the brain or the heart, the age. The brain or the heart scales as a power law of the body size, until about some point beyond which the brain saturates. But there are other internal organs, something that should surprise you. Your teeth grows proportion to your size. And there's a very nice power law which people have measured. Your stomach mass grows. Everything seems to scale very, very nicely across a huge range of creatures. This book by Prothero, it really changes your perspective of how mammals work. So we want the following feature to be evident in whatever feature. Let's say you have some pattern, and so the pattern could be a wave, so white, red, and so on. There's a certain body size, and there's a certain pattern size. The pattern is red and white. So at the initial scale, let's say 2 red and 2 white. But now, I could have had a case where the size of the white and the red remains the same as the creature grows in size. What happens? Put on more red and white. It's the same case with fingers. So, remain the same as you grew, so you put on. That is not what is happening in the living world. What's happening is the size of the features also scales with the body size. That's remarkable. Something that we all recognize when we look at children, right? There's an appropriate scaling of the every organ size as you scale the organismal size. But is this something at the level of creatures? Is this something at the level of tissues and organs? Or is this something that happens even within cells? Remember cell division that you on? A single cell divides into two cells, DNA duplicated, mitotic spindle forms and pulls the copied DNA to two daughter cells. People have wondered how this spindle structure would change its size if I had bigger cells. One can take extracts of such cellular material from frog eggs and put them in some microfluidic droplets, control the size of the putting in material there, and there is a of the here's, for example, the spindle length as a function of the droplet diameter that you're looking at. So we have a structure which is made up of polymer filaments inside our cell. Remember, a polymer. But somehow it has, the whole cell has grown bigger, and so this internal structure must also scale appropriately. How is this scaling achieved? This is a risk area we don't understand, but we have pretty good clues about how things work. The clue starts by thinking about how organisms develop. And that's why I tried to ask questions about not just looking at the adult size, but how the adult size is obtained from the So many, for example, the fruit fly, you keep a piece of banana in your home and there are these tiny black insects which come, that's the fruit fly. That's a fantastic organism for developmental biology, from which we learned a lot of physics and mathematics. Turns out the early divisions of this fruit fly make it into a very nice striped structure. So these striped structures are generated by the concentration fields of certain chemicals, and they have a very nice correlation with the adult body plan. So for example, the red structures eventually become the head, the yellow structures become the thorax and the legs and the wings and the trunk and so on. So the body plan, the structures that tell you which should become the head and the tail, is laid out very early in development. And very early in development, it's laid out by the concentration field chemicals. Biophysics has progressed remarkably well from the past 20, 30 years to an extent that we can actually experiments on living systems that at the level that you could do experiments on non-living systems. You do experiments on circuits and resistors. Now we can do experiments on living things as well as that. We can measure concentration fields very, very well. We can write down mathematical equations very, very well, and they seem to work. So it sort of gives credence to the idea that living systems are, at the end of the day, systems. They are very, very complicated. The scaling of these striped fruit fly, Drosophila. Even there, these earlier patterns, these exponential blue-colored patterns, seem to scale proportionately to the body size. So much so that the variant of these patterns, remember red, blue, red, white, scales almost linearly with the body size of the creature. Okay, I'm coming towards the end, and I would like to end with another movie of development and a problem that we looked at about how asymmetries in cell size come about during development. So this is C. elegans, it's a nematode. You'll find it in the soil. It's a wonderful creature to study many aspects of development. It's very nice that it's also transparent to white light. So you can basically see every internal organ of the creature just with the microscope. So there's this long worm which is a couple of millimeters in size, a bunch of eggs which are getting fertilized at various stages. You can actually look at one of the eggs, take it out of the mother, it's enclosed in a nice eggshell. And if you keep the pH and the temperature the same as mother's tummy, it doesn't even know it's outside mother. And it develops very, very beautifully. So this is the development of the nematode, and you start with a single cell, many cells dividing, and eventually a large scale mass of cells starts reorganizing. At some point of time, you'll see the emergence of a tubular structure, and this is real time. And in 11 hours, you start from a bag of cells, or a single cell, and in 11 hours, you have a fully developed worm coming up. 11 hours. It's remarkable that it achieves size, scaling, internal organ systems, proportionate scaling of all these internal organ systems. We don't understand the science behind this. What we understand to some extent is the first cell division. So the very first cell division of this creature turns out to be something which is asymmetric in the sense that the two daughter cells are different size. This kind of an asymmetry is achieved by a certain patterning of chemicals. Remember I told you concentration fields of chemicals give rise to certain patterns. These patterns can be accounted for very, very quantitatively. You can write down mathematics and the physics behind these patterns, and that seems to tell you how and where to divide the daughter cells and the asymmetry between the daughter cells. Okay, so I'll summarize that. So what I hope I have tried to convey to you is a sense that there is size and shape, and these are controlled across the kingdoms of life. And there is a huge variation in the size of organisms. They seem to obey scaling laws, in the sense of the various processes of life or even internal organ systems. And scaling laws are typically as power laws of the body size. There's a scaling which is emergent and it arises during growth, but it also is resilient and can arise during regeneration. The science of how these scaled patterns, size scaling patterns emerge naturally is typically by studying spatial temporal patterns of certain chemicals. And this is something that's ongoing, so we don't have a clear-cut picture of how these processes work. Alright, so I'll stop there. I want to thank my institute and my group, and I particularly want to thank my PhD student Jigyasa, who helped a lot in digging up many resources for this talk. I also thank the planetarium for inviting me. Thank you.
Thank you, sir, for this great talk. It helped. I'm sure it helped even the school students, to understand how scaling laws works from the most basic level and moving on to living systems, what governs the scaling laws and everything. So we are open to questions now. There will be 2 mics on either side. If you raise your hand, you'll get a mic and you can ask your questions.
So if the cells become a structure when they congruate, when the cells actually become together and become a structure,
yes,
do they communicate with each other?
Very much. Yeah.
And how actually they communicate? Through the chemical?
Yes. Communication is basically the cell releases some chemicals which goes and gets absorbed by the other cell, and there's a way to process the concentration fields of those chemicals to know, are you this size? Am I that size? Have I moved there? Are you ready to divide? We don't understand that language completely.
And those instructions, how to react to those, will be embedded in those?
Inside the cell, in the number of proteins, the gene expression patterns. Yes.
So I had a doubt in the regeneration part.
Yes.
So does the time the amputated part it takes to regrow depend on how many amputations and the size of the amputation? Just like
Yeah. I mean, surely. So planaria is special that you can amputate it in many different ways, and it seems to regenerate.
So if I take an entire planaria and cut off just the tail part,
it seems to regenerate. There's some minimum cell number, few hundred cells or something like that, and beyond that it's almost completely. It's a remarkable thing. Just a few hundred cells can recapture an entire patient.
So it also depends on which species is getting amputated.
That's right, and which organ.
Okay. So
not all species can do all organs.
So what about the others?
I think I pointed some of them. Newts can regenerate limbs and fins and hearts and so on.
So that also depends on how much of the limb
Very
is
good. We are also capable of regeneration. Skin, for instance, is something that we all get scratches, right? And that gets regenerated. But not limbs, not hearts, not brains definitely. Nowhere. But there are, so that also depends on the complexity of the creature. Simpler the creature, probably it's easier to regenerate. The more complex the creature, it's harder to regenerate.
Thank you.
Will zebrafish die?
Will zebrafish?
Dies?
Yeah, sure it does. At some point of time it will die. If you don't give it enough food, it will die. Just like us.
Because
so all creatures need food. One way to kill a creature is don't give food. That definitely kill it.
Because you said that the heart, if a heart will be
yes.
cut, at that time it will grow again.
Yeah, it can regenerate, provided you give it food. If you keep giving it food, it can regenerate its own dead heart. That's amazing. No?
Why can't all organisms regenerate its all parts of the body?
I didn't get it. Why they regenerate?
Why can't all?
Okay. We don't know. That's the best answer. Like I was answering, simplest feature, it's probably much easier to regenerate the entire organ system. The more complex the feature, like us, for instance, it becomes harder and harder to regenerate the entire body. You probably can regenerate some tiny part, but not the entire body. The area is very, very simple, extremely simple feature, and somehow has the super capability to regenerate its entire body. Why it does that, how it does that, can we use it to understand other things? We don't know yet.
But in the small creatures, the ratio speed is very high to regenerate its organ.
It depends on the creature. I showed you something for planaria, right? It's a few days, few tens of days. Depends on the creature, depends on the environment, how much nutrients you provide. But the fact that it can regenerate is something that's useful.
Any other question?
Is there a special cells for regeneration?
Sorry, can you say that?
Is there a special cells
So I
for
didn't
regeneration?
want to go there. Yeah, it turns out that stem cells are the primary determinant for regeneration. So the more stem cells there are, stem cells are a particular class of cells which have not differentiated. They have not become specialized to eye cells, head, tail cells. They can become any other kind of cells. So the ratio of the number of stem cells to non-stem cells is a rough measure of how good the regenerative capability you have. The myriad seem to have a huge number of stem cells. And so therefore they can regenerate any organ that is lost.
Can't we take cells which can be
Which can?
regenerated body parts?
Can't we?
The stem cells.
No, we do have stem cells within us too. But they are very, very specialized and they are already located in different parts. The process by which they can be used to regenerate any lost limbs, lost parts, doesn't seem to be so prevalent in higher order creatures like this. Again, it goes back to the same point that there are simpler creatures. There it's much easier to modify.
Sir, when the cell starts to grow from the first stage, if certain elements are not provided or they are missing, what are the factors would affect, what would happen in the cell?
For example, think of the first cell division. Suppose you don't provide enough food. The food is eventually converted to forces which will actually make the cell divide into two pieces. Suppose you don't give enough food. There's no energy. So therefore the forces that are generated are weaker. And so the cell can't divide. So that's one drastic example. So if the first cell division itself doesn't happen, forget the rest. Even things like cell movement, cell deformation, cell shape, everything needs energy. And so one way you can say is that, let's say you put out energy, the input, the nutrient, the food, you will drastically affect the subsequent development. Does that make sense?
If everything is provided, just once certain single or two.
If you remove one molecule, 10 molecules, 100 molecules, nothing will happen. It can take care of things from the environment. But there's some critical number beyond which some vital structures will get disrupted. That's when it stops.
What about our internal organs, sir? Do they regenerate during any surgery or anything like that?
Skin definitely does. But then the internal organs are also much more complex. One of the best studied creatures is mice, like I said. Mice is capable of regenerating the entire liver.
What about the humans, sir?
Not much. It would be fantastic if we could, but we don't know.
After a surgery, I'm asking, sir. After a surgery.
I mean, that's one of the main things. If you could find regenerative capabilities, any wound healing, any surgery that happens, immediately you could regenerate the entire part. We don't seem to have that capability.
Sir, is it very tiny number of species that they undergo to regenerate?
Yeah. Very few.
I mean, for example, the mice, I mean, there may be, I mean, if you take a size or scale, there may be some species which may be very similar to that. But There are many actually. Why actually only mice can undergo?
We don't know the answer to that. What we find is that mice can do this job. I mean, like I said, every creature can regenerate. But the question is, how much can it regenerate? Can it regenerate everything? Or some parts? Or very tiny parts? These are the creatures which have a maximum regenerative capability. They can regenerate them more or less the entire body part. Why is it that they are actually like that?
And the last question from me. The force and the energies and all those things involved in this are very, very tiny.
Yes.
How
Tiny
we can
compared
measure?
to
How we
what?
can we measure? Or
Yes, we
estimate?
can measure. There are plenty of mechanisms to measure. You can do direct measurements using microscopy, force microscopy, atomic force microscopy. Or there are indirect ways to measure flows and deformations, and from that back calculate the forces. The forces are typically of the order of piconewtons in a cell, for instance.
Thank you.
Sir,
Yeah.
so if people see interesting things like this regeneration and all, they become happy and they want to replicate in our
I
life.
don't know if the planaria is happy. I don't know what that even means for planaria, but go on. Yeah.
So have the people tried to replicate regeneration in human also?
That's exactly what she was asking. There are some parts that can be
Taking
regenerated.
the part from...
No, not. Maybe something else other than regeneration.
So can't we adopt these types of cells and tissues
To what?
humans?
No. I mean, you're saying take planaria cells, put it inside you, and maybe you'll become a planaria. No, there's a serious incompatibility of chemicals and genes, so that won't work.
can we put more stem cells to generate more parts?
No, it's not just a question of just put a lot of stem cells. But the stem cells must also be clearly incorporated in your body. They must set in very nice composition with the remaining parts. They should get enough food. If you just put lots of stem cells, you might become completely stem cells. Muscle cells are not there. You can't move your hands or legs. What do you do then? What's the point of having completely muscle cells, stem cells? You need other kinds of cells as well to do your functional things. Now, what proportion is to be stem cell? What proportion is to be non-stem cell? And so therefore that sort of regulates regenerative capabilities. That seems to be highest in planaria. I'll remove the regeneration slide. Everybody seems to be asking regeneration.
Sir, one question. Sir, I think when one of the students asked, will the zebrafish die?
Yeah.
That's the first question.
Yeah.
Suddenly I thought that the student was referring to mortality in some sense. Because if you keep regenerating each and every part of your body, you can live forever. So my question was, in the evolutionary timeline, what stopped evolution in some sense, or some organism from living forever?
No, evolution has not stopped.
No, as in like, why can't an organism live forever through regeneration? What stopped?
I mean, planaria seem to live forever.
Sorry?
Planaria seem to live forever.
Okay.
They don't seem to die. I mean, you keep giving it food, and they're happy to keep growing. And you chop them up, they'll regrow.
Okay, but when it comes to higher organisms?
Yeah, that's when the regenerative capabilities go down.
Okay.
By the way, evolution has not stopped. Evolution is going on even now.
Yeah.
Only that on our time scale we are not able to see that very well.
Okay.
Sir, in human beings, these days we are hearing more of this transplantation.
Yes.
It may be liver, heart, even now. So now it's not simply to place it in that original place, but it has to develop the other blood vessels,
Exactly.
and there should be a contact with the other organs also as well.
Exactly.
So is it not a regeneration?
Maybe, maybe not. Those would be more like grafting. There have been plenty of experiments in grafting. This goes back a little bit of history, about 100 years ago. People were studying many examples of frogs. In the early stage of the frogs, they would chop off the limb of one frog and put it on another frog. Now what happens? Does it grow an extra limb? Let's say you take it from a small frog and put it in a bigger frog. Does it make a bigger limb? Does it make a shorter limb? There seems to be a huge variety of things that's happening there. This is some form of the compatibility that you're asking. If I take one piece from here and put it there, should it reconnect to the existing structure?
It is connecting because of the transplantation is done. they can see the person growing and doing all other activities that happens in his lifetime
That is correct.
or her lifetime.
That is correct.
So it has to be done. It's not like fixing with a wire and just leave it.
No, it sort of gets into compatibility with the existing structure.
Yes.
I agree with the blood
It
vessels
has
and
to
so on.
coordinate with the other
Would that be
functions.
regeneration? Maybe, but that's not what's used in this sense for these creatures. I'm not aware.
Okay, thank you.
How many times can it be regenerated?
I can't hear you.
How many times? How many times can be regenerated?
How many?
Times. Only once it can be regenerated.
No. Anything into infinite number.
Infinite.
I mean, there's no documented limit.
Sir, I have a question. So, liver in mouse.
Yes.
It seems
Everybody's regeneration, fine.
no, it's kind of very specific. Is there any idea why?
I don't. Not another reason.
Many animals or hydra itself dies by age or its age.
Like I said, if you keep giving it food, it seems to survive forever.
Means now one hydra will live 1 month. Then if you cut its part, then which is older?
So it doesn't seem to care. It just seems
Two
to.
hydras, which is older?
Yeah, it seems to regenerate. It doesn't seem to care. The older one or the younger.
Sir, here, sir. Yeah, so adding, I forgot to ask the same question, similar question to what ma'am just asked, like why liver in mice particularly. So is it because, like, let's say, rats eat very bad things and liver damage?
You can make up a story.
Like, have you particularly looked into some reasons of why certain parts in certain animals?
I have definitely not. I don't work in that area.
Okay.
But you can make up some story.
Okay.
But there isn't hard evidence as well.
Okay.
Any more questions? So then we'll end the talk here. Thank you everyone for coming here today. I'm sure you enjoyed the talk. We have 2 more lectures as part of Dasara Lecture series, tomorrow and day after. So they're very interesting talks, and I hope you guys visit us, even tomorrow and day after. Thank you again, sir. So we have coffee and biscuits outside. So